I quickly do like an overview of the cardiac cycle, and um, I will talk a little bit. Want to talk about like the different things that affect cardiac output and stuff, just because that makes the picture a little bit clearer. But anyway, all right. So let's start from like the very beginning of like what's happening to the heart. So let's say this is our heart. Gonna do like a very rough diagram of it. I guess actually here, like here, down. Yeah, here. Okay, awesome. So this is your atrium, this is your ventricle, this is like the atrioventricular valves, like the AV valves. It's very badly drawn, but like AV valves. And this is your semilunar valves. Semilunar. Okay. Both sides have semilunar, but the left side is called the aortic valve because it goes into the aorta. Um, yeah. So both happens at the same time. So I'm just gonna like work with the left side or something, you know. Yeah, because it's like the same. All right. So at first, what happens? Is the AV valve open? Like at the very beginning when things are gonna fill in, right? AV valve. So this fills. Yeah. So yeah, we're gonna start. Let's start with like the AV valve open, just because we're gonna come back to it. It's a cycle, so you can pick wherever. But let's start with AV valve is open, which would allow blood to what passively fill both the chambers, atria and ventricle, let's say. Okay. If they are relaxed, then diastole. So both are relaxed at this point. So that is fine. So just what would happen to the blood volume in the ventricle? Yeah, the volume is increased. What about pressure? Anything happen to the pressure? Yeah, nothing's happening to the pressure. No one's contracting. And then what would happen? There would be like atrial depolarization. That's like the P wave, right? Okay. And that causes, yeah, atrial, oh, sorry, or like basically contraction, right? Okay. So they would contract, which would kind of like not do much, but like squeeze the last bit of blood into it. Okay. So squeeze last bit of blood from atria to ventricles. Okay, and that would kind of increase the pressure like slightly. Okay, very slightly. Is that okay? Okay. Just because they have like squeeze it. Okay. Hmm. Which is not true. That's a very good misconception. I think a lot of people have actually. So that's a very common thing. Yeah. So that's why I like starting with like everything open, with only like the semi-lunar valve closed, obviously. But like, and both are filling at the same time is like a good idea. Cool. And then the atria has contracted a little, so squeeze the last bit of blood. And then what happens? The pressure change, the valve was snapped closed when the pressure at the bottom is like here greater, right? Because they would like push the blood up to like close it. Yeah, but now the ventricle hasn't contracted yet. So that wouldn't happen. That's like when you're squeezing it, right? The ventricle didn't start squeezing yet. What makes it start squeezing? Yeah. So there would be like ventricular, that would start when they start to contract. Yes. So ventricular depolarization, that's the QRS wave. Cool. And that makes ventricle contract, which would do what to the pressure? Yeah. And does anything happen to the blood volume yet? When it's still like isovolemic. So blood volume would not change, so it's isovolemic. Okay, fine. Now the pressure has increased enough to open the valves, right? What valves? The AV valves closed quite a bit earlier. That's why you have isovolemic. So isovolemic contraction would start when the valve has closed. So blood cannot go back up. So when contraction starts, that's when like the valves close. So I can like maybe I can like write it here. Yeah. Okay. Closes AV valve. Would have been great if I had used a different color for things, but I did not. Anyway. All right, so we have closed AV valve, that's fine. So now you have isovolemic contraction. I'll just write contraction. Okay. And the contraction becomes strong enough to open the what valve? Yeah. Semilunar, or if it's like on the left side, then aortic valve. That would send blood to where? Yeah. Yeah, to the aorta. Yeah. So that would be just like blood would be ejected out of ventricle, wherever it wants to go. Okay, so what would happen to the blood volume? Yeah, in the ventricle, the blood volume would decrease. Cool. And then let's say we go back to our electrical signals. What's happening? What's going to happen now? Actually, probably happened while here. So like probably right here. During this period, something happened with the electrical signal. Ventricles like repolarized, right? We said repolarized. Yeah, that happened already. So now the ventricle would repolarize. Ventricular repolarization. Sorry? Yeah. That's your T wave, right? Okay, awesome. And just like a side note, the atria also did repolarization, but that was during the QRS signal, so it was like masked. Yeah. That's why we didn't see. Okay, cool, awesome. All right, so if the ventricle is repolarized, that means the ventricle would relax now. If the ventricles relax, then what would happen to pressure? Lowers to the point that aortic or semilunar valves close. And if that happens, then both of the valves are closed now, which means you have blood volume not changing, which is the ISO. Cool. And then it becomes so low that the AV valve open again, and it's like a cycle. Cool. Yeah, but it's like putting it together. And this would be really helpful for you to make the Wiggers diagram with it. Wiggers diagram. That's like when you put like all of it together, like the QRS, like the ECG thing that we learned, then just how pressure changes with time, that and like volume changes with time, like all of it in one diagram, which is kind of cool. So I think the professor will teach it, probably, or we'll probably do like a group problem within there or something. Okay. Great. That was nice. Um, cool. The other thing I wanted to very quickly go through, just because that would also clear stuff up, or put things into context, is how mean arterial pressure changes. So do you know what mean arterial pressure is yet? Okay. Okay, cool. So cardiac output, you learned is like SV and heart rate. Awesome. So, just giving you context, where would those go? So, mean arterial pressure is like what's like the overall artery pressure, and that's imp yeah, but like they average it. So that kind of gives you an indication of what blood pressure is. Like, you know how you hear like blood pressure of people, whatever. That kind of gives you like a very good, like rough estimate of like blood pressure in general. And that's really important because your brain wants to maintain that, like keep that stable, which makes sense because you don't want the blood pressure to like increase too much or decrease too much. Then if it's decreased too much, your organs are not going to get blood. If it increases too much, it might damage like tissue, like delicate tissues in your lungs and kidneys, nephrons, and stuff. So you don't want to do that. So your body wants to maintain it. So how does it maintain it? It can manipulate the other like the components of MAP. So cardiac output is one of them that you learned, and cardiac output depends on stroke volume and heart rate, which is also you learned, and this could be manipulated by the brain. The other thing that you didn't learn yet, but you will, is the total peripheral resistance that you will learn. So I won't talk about that today. But let's just talk about the stroke volume and the heart rate. So the heart rate could be manipulated by both like the sympathetic and the parasympathetic stuff that we kind of learned before. So sympathetic, parasympathetic. If you remember, the sympathetic would release norepinephrine onto beta one, which yeah, of what cells? Sorry, beta one. Beta two is for it's gonna come for the TPR thing. We'll talk about that later. But like yeah, beta one on what cells? This is heart rate. Not all like if for heart rate, then yes, the AV node. So that's the autorhythmic cells. Yeah. But if it was for stroke volume, that would be the cardiac contractile cells, the ventricular cells. Yeah, we'll come to that. Okay. So rhythmic cells. Yeah. Okay. All right. Okay, no problem. But because you did learn these in the previous somewhere. Anyway, so that would cause the heart rate to increase. Simple. Okay. Parasympathetic was the opposite. Do you remember what neurotransmitter was being released by the postganglion neurons of the parasympathetic? Yeah. And that was binding to what receptor? Perfect. And what do they do eventually? It is also on the autorhythmic cells, right? Well, what do they do? Perfect. Yeah. Yes, actually, that's a very good idea to do that. So let's say I'm gonna just put it down. Okay. So beta one is like what do they do? Do you remember what G protein was that were they part of? Like, sorry, connected to? GS. What does GS do? Yeah, yeah. So this camp, increase in camp actually can interact with the iFunny channels that you have learned about. Yeah, yeah. So that makes like higher action potential easier. So that makes heart rate easier. Okay. Um, for the muscarinic, did they talk about it? Yeah. Yeah, there is a specific one. Did you have to learn that? Okay. Okay. Oh yeah, I think there was in the slides. Wait, hang on. I can go back to the slides and find it. Yeah. Um, yeah. Okay. That could be helpful. So the last one. Oh, not 11, 10, right? Oh, here, I think. No, this is for the stroke volume. Muscarinic. It didn't say. Um, yeah, I also don't think that you have to know it because the group problem question you got was very generous about like just knowing that it was connected to GS and the hint they gave you about the seed camp and the affinity channels kind of like gave you the answer. Um, don't think you have to learn that. The the one that is present about the phosphorylation, that's for stroke volume. For cardiac muscle cells. So that's different muscles, like different cells, not the autorhythmic cells. They both look different with the action potential, if you remember. Like the autorhythmic ones kind of look like this, like, and the cardiac one looks like this, right? Yeah. So the mechanism is different. So this was the mechanism for the beta 1 receptor for autorhythmic cells. If we go to this side about the stroke volume, then another key thing to remember that people often mistake is the stroke volume of the heart, like by the heart itself, only has connection with the sympathetic, not parasympathetic. So parasympathetic can't make the heart contract harder, but the sympathetic can, like are less hard or whatever. Yeah, exactly. So yeah, yeah. Wait, slide what? 18, you said? 16? Yeah. So autorhythmic cells. Okay. Okay, perfect. So, okay, that's all you have to. Okay, cool. All right, that's fine. Like, that one's actually a little later. Maybe she'll talk about it today. Maybe. Okay. Okay, that sounds about right. Okay, then you know what? I'm gonna like skip it. She's gonna talk about this part. Then yeah, how it affects the contractility. But let's talk about this part then finish it up. So the parasympathetic. Oh, okay, sure. Yeah, I'm fine with that. Um, okay, so continuing. So parasympathetic acetylcholine binds to muscarinic receptor, which then activates GI, which then does what? This was M2 type. Okay. GI, so that really like causes less activation of PKA. Yeah. With the G protein. So the alpha one is the like the key one that does stuff. So it decreases CMP that decreases phosphorylation of like protein kinase A. And the beta gamma we that really like depends on different times and I feel like maybe it's not important here or maybe it is. Like that's for oh yeah. Okay, so that's a channel, a potassium channel, and they make it like open it, which would hyperpolarize the cell. So there are two ways that this is happening. Um, so for the PK part, it would cause like less phosphorylation of the T-type calcium channel. But for the beta gamma part, they're going to cause efflux of potassium, which would make the cell hyperpolarize. Do we have to know more details? Probably not. Do we have to know if this binds to GERK? If you can remember that, like, feel free. Um, yeah, so that would basically make Okay, and this one, the pK would like unphosphorylate, I guess, or dephosphorylate. Yeah, yeah. Oh, yeah. Actually, that's a better one because that would doesn't mean like, oh my God. Okay. Yeah, phosphor. Oh my God. Of the T-type channels. I mean, both of it eventually makes it like harder for action potential to start, so lowering heart rate. Another key thing I've kind of noticed people do a lot in writing answers, they don't complete the thought. They just like answer part of it and they don't connect the dots to like what happens at the end. So it would be a good idea to like always do that because your main mark is being carried at the end, like how you connect it. So even if you don't mention a few tiny details somewhere, we the professor is pretty lenient about it. But if you don't mention what happens at the end, it's like I don't know if you know what happens, you know. So, yep. Okay. Is that okay?